structure in visual cognition. So one of my big interests are multi-scale interactions. I think that these interactions give rise to complex nonlinear structure that can be useful for helping us find and explain observable behaviors. So, like most psychologists, I am interested in human behavior, which happens at a particular scale of time and space. Humans are typically nested within some kind of environment that's at a larger scale of time and space. Nested within the human are obviously organs and sensory systems, perceptual systems, and within those systems are nested cells, genomes, molecules, and so on. And across all these different scales, there are interactions that kind of go all sorts of which ways. And these interactions give rise to this nonlinear complex structure. So, for example, we can search for fractal structure in biological signals. So by fractal, I mean these scale invariant, self-similar patterns in these different signals. And we can use these to predict different facets of behavior. So take, for example, postural sway. If we were to record postural sway during some kind of cognitive task, we could search for structure in that signal. And in fact, in some of my previous work, I've done just that. During a stimulus intake phase of individuals estimating geographic slopes, either visually or haptically, we were able to measure the signal for multifractal structure. And we used this multifractal as an index of the complexity of the signal. And this index went on to predict their perceptual responses, their cognitive responses, and also matching across the different modalities, suggesting that the structure sort of weaves together short and long time scales and spatial scales to be able to produce this behavior. We can also look for fluctuations in the pupil response when we're thinking about how the visual system sort of habituates to multisensory stimuli. And in this case, we might look for higher order polynomial structures. So here at UW, we've asked some very young children to watch these multisensory displays. Half of them are neurotypical, some of them are young children with autism. And during this experiment, we tracked their vision, we tracked their eye movements and their pupil fluctuations to generate these time series. And what we're looking for is habituation of the pupil, that is decreasing pupil over the course of the experiment. And to simple visual displays such as this optic flow, we can see differences in the structure of their habituation response, such that the neurotypical children show this sort of linear decrease in pupil diameter, but the autistic children show this quadratic structure where they increase and then decrease over the 2 halves of the experiment. In more complicated dynamic scenes, we see both sort of quadratic structures in their habituation responses, and they tend to differentiate towards the second half of the experiment. But what's interesting is that we can estimate the slopes of these predictive models to find the rates of change in the habituation response, and we see that neurotypical children habituate much quicker in this case. So in this respect, it's the structure that is sort of being produced by these multi-scale interactions that I think is useful for us to search for and try and utilize to explain these observable behaviors. So with that, I want to thank my advisors here, Gabriella and Sarah, and the rest of my collaborators, and I'm happy to take your questions. Time for a question.
I'm just curious what does that quadratic structure mean? I mean, is this like indexing? This is indexing arousal over some period of time, along those lines. Like, well, why do you observe that quadratic structure?
So these children are sort of watching the same stimulus over and over and over and over again, and we're expecting the pupil to sort of show decreases as a function of the length of the experiment, as the number of trials.
Yeah.
And so what we were expecting were just both groups to sort of habituate at the same rate, but we see this difference in the autistic children are actually increasing towards the first half of the experiment, suggesting that maybe the stimulus remains novel for longer. It's an idea of like a failure to update predictive prediction errors. So we think it's interesting that for the very simple stimulus, The neurotypicals don't show any kind of increase, but the autistic children do.
So the non-monogenic nature might be that you're sort of realizing that you're not doing very well and modeling it for a while.
Yeah, so they're not updating their predictions so that it doesn't become sort of stale, I guess.
Okay. Cool, thank you.